Hello, my name is Emily, and today we're going to be talking about virus basics. First of all, what is a virus? A virus is an infectious particle that consists of some genes packaged in a protein coat. It lacks structure and metabolic machinery, so it's very simple and small compared to eukaryotic and prokaryotic cells. Viruses can also cause many diseases. And over here is a picture of some viruses. Characteristics. They cannot reproduce or carry out metabolic activities outside of a host cell. Biologists classify them as nonliving, and more specifically, they are in between life forms and chemicals. Here's another picture of viruses under a microscope. The structure. A virus is a nucleic acid surrounded by a protein coat. The smallest virus has a diameter of 20 nanometers, which is smaller than a ribosome, while the largest has a diameter of 1.5 nanometers, which is still barely visible under the light microscope. Some are surrounded by a membranous envelope, but not all. Over here is a diagram of a virus, and it has the labeled parts. Genomes. A genome is an organism's complete set of genetic instructions. They may consist of double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA, but it depends on the type of viruses. They can also be classified into DNA virus or RNA virus, but depends on the nucleic acid that makes up its genome. Organization. Viruses are usually organized as a single linear or circular molecule of nucleic acid. Some genomes of viruses consist of multiple molecules of nucleic acid. The smallest viruses has only 3 genes in their genome, while the largest has 2,000 genes. And if you compare that to bacterial genomes, they have very few, because bacterial genomes contain about 200 to a few thousand genes. Capsids. A capsid is the protein shell that encloses the viral genome. The shape depends on the type of virus, so it can be rod-shaped, polyhedral, or more complex in shape. They are built from capsomeres, which is a large number of protein subunits. Over here is a diagram with the labeled part capsids, and underneath is another diagram and another labeled part. Envelopes. Viral envelopes are the membranous envelope that surrounds the capsids of influenza viruses and many other viruses found in animals. They are derived from the membranes of the host cell and contain host cell phospholipids and membrane proteins. They also contain proteins and glycoproteins of a viral origin. And if you don't know, glycoproteins are the proteins with carbohydrates that are covalently attached. Structure diagram. Over here is a diagram of a naked virus compared to one with an envelope. So they both have capsid and genome, but over here it has an envelope which is labeled. Capsids and envelopes. Some viruses carry a few viral enzyme molecules with their capsid. Many of the most complex capsids are found among the viruses that infect bacteria, and over here you can see another diagram. Replication process. Because viruses lack metabolic enzymes and equipment for making proteins, including ribosomes, they replicate within a host cell. Each particular virus can infect cells of only a limited number of host species, which is called the host range. They are usually identified by a lock and key fit between viral surface proteins receptor molecules on the outside of cells. Some features include the used equipment and small molecules of its host cell to replicate. The simplest of viral cycles is a parasite that is a DNA virus with a capsid consisting of a single type of protein. Over here is a diagram, and if you follow the arrows over here, they show like the different steps. But here are the different steps to the cycle. First, the virus enters the cell and it gets encoded. Then it releases viral DNA and capsid proteins. Secondly, the host enzymes replicate the viral genome. Third, these host enzymes transcribe the viral genome into viral messenger RNA, and the host ribosomes make more capsid proteins. Lastly, the viral genomes and capsid proteins self-assemble into a new virus particle, and then they exit the cell. And here is the picture version of what I just said. The numbers are the different steps. And you can pause the video if you want to look into this diagram in more detail. And that's it. Thank you for watching. I hope you learned something. Please follow our Instagram and subscribe to our YouTube channel. Bye.